All right, we're getting started, okay?
Sure.
Hi, everyone. We're just waiting for folks to log on. So we'll just start in about a minute or so. Okay. All right, why don't we go ahead and get started? Thank you so much for everyone who's able to join us today to talk about Cockayne syndrome and drug repurposing. We really appreciate all of our CS families that are joining us from around the world, all of our fellow researchers, and a special thank you to folks joining from Leiden University Medical Center and Charles River Laboratories, who are our research partners on this project. Let me introduce myself and give you a little bit of background, a high level overview on what we're going to be talking about today, and then I'll turn it over to our esteemed panelists, and we can ask questions and uh, you can ask Q&A along as we go along. You can type in your questions and we'll address them at the end. You can also raise your hand. There's an option to raise your hand at the bottom, and we can unmute you if you want to ask your question instead of typing it at the end. Um, my name is Kaur, and I am the founder and chair of Riaan Research Initiative, which is a nonprofit organization based here in New York City, with a mission of getting treatments developed for Cockayne syndrome. Um, we were founded after the tragic diagnosis of my 3-year-old son, Riaan, who was diagnosed at 15 months, so in March 2021 with CS type 2. We were devastated, as is every family that gets this diagnosis. 3 months after he was diagnosed, we decided to launch, or we launched Riaan Research Initiative, because we wanted to do more to see if we could play a role in getting treatments developed. You know, this disease is horrible, it's lethal, there's a lot of complications, it's very complex. And at the moment, there are no approved therapies. One of the programs that we explored is called drug repurposing. So just to give you a little bit of sort of, let me step back for a second. When we think about traditional drug development, just getting a drug out into the market from idea to clinic to being able to prescribe it at a Walgreens or a CVS or your local pharmacy, it can take up to 10 years. It can cost $1 billion. And quite frankly, when you have a disease as ultra rare as Cockayne syndrome in such a small population, there's not really a lot of incentive for pharmaceutical companies to develop a drug from scratch to treat this disease. So we looked into what's called drug repurposing. Now, essentially what that means is, let's not reinvent the wheel. Let's look at all of the drugs that are currently in existence today, FDA approved drugs, drugs that are used in clinical trials, and see if we can use them for a different indication than what was originally intended. So maybe the best known example of this, a successful or perhaps even uplifting example, is a drug known as sildenafil. Sildenafil was developed by Pfizer to treat hypertension or high blood pressure. And they quickly realized that it was also causing erections as a side effect and could be used to treat erectile dysfunction. So it was repurposed for the treatment of ED and is now known as Viagra. So I'm sure everyone's heard of it, but that's sort of a common example that's often used as a successful drug repurposing program. Now, Cockayne syndrome, as I mentioned, is a lot more complicated than that, but the basic premise is the same. Are there drugs out there that could potentially halt some of the neurodegeneration that we see in our children, that could alleviate some of the symptoms? There's over 5,000 FDA approved compounds. Could one of them work? And if so, how do we figure out if it works? So we contacted Martijn, our organization, back in October. Dr. Martijn Luijsterburg, who is joining us from Leiden University Medical Center. He runs a DNA repair lab. He is one of the foremost experts on the molecular aspects of Cockayne syndrome in the entire world. we contacted Martijn and he said, listen, can we develop some sort of test system to get at the heart of the neurological problem of CS? We know that it's a DNA repair disorder, but we also know that fixing the DNA repair issues alone will not address the neurological problems that our children face. So Martijn came back with this extraordinary idea, which is, well, why don't we develop an assay using a early transcription factor gene called ATF3? He said that there had been a paper that was recently released about it. And essentially, what ATF3 does, and you'll learn more from our presenters, is it's a gene it's a stress protein that essentially is always activated in children with Cockayne syndrome. In fact, it's even considered a biomarker for the disease. And it's linked to other genes that play a role in neurological and neurodevelopmental processes. So when it's activated, it could potentially lead to symptoms that cause microcephaly, that cause primary or motor neuron or motor sensory neuron delays, things of that nature, symptoms that we see in children with Cockayne syndrome. So essentially, the nuts and bolts of our program, and it also disrupts transcription processes. The nuts and bolts of the program are, well, can we build a test system, an assay based on ATF3, and then can we screen all the drugs that are out there to see if we can find drugs that can inhibit ATF3, and could that potentially help our children? Can that sort of stall the neurodegeneration that I had mentioned earlier? So I'm going to now turn it over to Martijn and Paula Meer, who I'd like to introduce. She is a PhD student in the Department of Human Genetics at Leiden University. She works for Martijn in his lab, and she's been pivotal in this project from day one. So Paula, I'm going to go ahead and turn it over to you to sort of explain in more detail. And like I said, if you have any questions, feel free to ask them as we go along, type them in, and then we can address them all at the end. Thank you so much.
Well, thank you, Jo, for the kind introduction. I'm Paula, indeed. I'm a PhD student in the lab of Martijn. And as Jo already clearly explained, in this lab, we do research about Cockayne syndrome. And we do this on a very fundamental level. So we really try to figure out how the disease works on a molecular level, cellular level. And we try to understand what really happens in Cockayne syndrome. And indeed, now together with Riaan Research Initiative and also Charles River, we're setting up this project for finding maybe a potential treatment for Cockayne syndrome. And today I want to explain to you the approach we're taking with this and how it is going. But before we are going into that, I want to give a little bit of an introduction into cell biology in general, so that everyone is on the same level and everyone can hopefully at the end of this clearly understand the biology of Cockayne syndrome and also how we approach this repurposing screen for Cockayne syndrome. So let me start at the beginning. Our human body consists of a lot of cells, many cells, and also many different cell types. Here you see 3 examples of these different cell types. For example, you have red blood cells, you have nerve cells, you have skin cells, and like that, there are many other cells, and each type of cell has a different function in the body, and all those cells together form your body and make sure your body stays functioning and stays alive. And you can see already every type of cell with its different function also have a very different form, so a very different shape to execute this function correctly. And all these different cells do have something in common, and that is the DNA of those cells. The DNA is in every cell in your body. In the nucleus of the cell, there is this DNA, and if you zoom in on that DNA, you see this very long strand, this very long molecule, and actually it's 2 strands that are really wound about each other, around each other. And these 2 strands, in between of those, they are connected via nucleotides. And there are 4 nucleotides. We call them A, T, G, and C. And these 4 nucleotides are binding together. And here the A is always binding to the T, and the C is always binding to the G. So they're always opposite of each other in that way. And all those 4 nucleotides form a very long code throughout this whole molecule, throughout this whole strand. And as you can see, it's super long because it's folded very tightly in this molecule, and only when you zoom in like this, you can see the nucleotides. So these form a very long code in the DNA with these 4 nucleotides. And why do we need this code in our cells? Well, on the DNA are actually in this code the instructions for the cell to make proteins. And you can see this as a sort of cookbook, a sort of book full of recipes. And these recipes form then the instructions to, in the end, make, well, for example, this pie. And that will then be the protein. So every gene contains instructions to make a protein. And you see already in between there's something else. There's this mRNA, that's the messenger RNA. And you can see that as a kind of grocery list in this analogy we have here. So that contains all the ingredients, that copies the ingredients from the cookbook, from the instructions, and then make sure that from that ingredients the protein is made. So the pie is made here. So indeed, this messenger RNA is a messenger that copies the instructions from the DNA. And with those instructions, the protein is made in the end. Well, why do we need this messenger in between? That is because, as I said, the DNA is in the nucleus of our cells. And the protein builder of the cell, in our analogy, that will be the cook. That is not in the nucleus, but that's outside of the nucleus in the cell. So we need this messenger to bring the information, to bring the instructions from the DNA to the cook, to the protein builder outside of the nucleus. So that is why we need the grocery list, we need the messenger. And then if you then look at this very long DNA molecule that we were looking at with this very long code, on this code there are many, many genes, and every gene encodes for a protein. So one gene has the instructions to make one protein, and the next gene has the instructions to make another protein. And from each gene, this messenger is first made, and then that is brought outside the nucleus to the protein builder, and the protein is then made. So every gene encodes for a different protein. And here you see the summary of that, and this is the consensus of whole cell biology, I would say. DNA has the instructions for proteins, and in between this messenger carries these instructions to make the protein. Why do we need all these proteins? Here you see all shapes and sizes of different types of proteins we have in our cells. And this is what that would look like if you really look at a cell. So you see many different types of proteins. For example, you see proteins here that form some kind of skeleton in the cell to really make sure the cell keeps the correct shape. You see proteins that are on the membrane of the cell that make sure there's transport of, for example, nutritious compounds that the cell needs. You have proteins that make energy for the cell. You have proteins that perform some kind of chemical reactions that are necessary for the cell. So in all those different types of ways, each protein is needed for the cell to stay alive and to perform its function in the body. And all those different proteins are encoded on all these different genes in the DNA. And now I want to zoom in on this process that's called transcription. So where from the DNA, the instructions, the code is copied onto the messenger. And that process is also performed by a protein. And here you see in a little video what that would look like. The red strand here is the DNA, and you see this blue protein, that is the reader protein, and that reads the DNA. It races over the DNA and reads it while it's doing so, and while it's reading this DNA, it makes this messenger, and that's the yellow strand coming from the top. And this is the messenger that then brings the instructions in the end outside the nucleus to the protein builder. And you can see already this reader protein is going super fast over the DNA. So you can also imagine that it's really important that the DNA that is going over, that it's reading, that remains intact, that it's not damaged or anything, because if it will be damaged, then also in the mRNA there might be a mistake in there, and in the end there might also be a mistake in the protein that's supposed to be produced from these instructions. And it's quite a challenge for the cell to keep the DNA intact, because there are many different ways for DNA to be damaged in the cell. For example, sunlight can damage DNA. That's what happens when you get sunburned. But also chemicals or different types of radiation, but also just making energy in the cell, which is something that happens the whole time in all your cells, that produces some compounds that also damage your DNA. So DNA is continuously damaged in all your cells, but luckily your cells can repair that damage so that the DNA remains intact. Well, now I want to talk about one specific type of damage that is caused by sunlight. And what that type of damage looks like is like this. Normally, of course, the nucleotides bind to the nucleotides on the opposite strand. But when DNA is damaged by sunlight, instead of binding to the opposite A, this T now all of a sudden binds to its neighboring T. And when you look at that in 2D, that would look like this. You see this kind of bump in the DNA. And if you now think back to the video we saw with the reader going over the DNA very fast to make the messenger, that can cause a problem, of course. And indeed, if you look at the reader, you see a little cartoon of it where the reader is the green protein you see here making the RNA. And it's going across the DNA strand. But then if it encounters such a DNA damage as this star here, then the reader cannot cross that. It cannot come over this little bump. And it's stalled on the DNA. Well, that's a big problem for the cell if the readers are stalled on the DNA. Luckily, we have a mechanism, or most people have a mechanism, to repair these kind of damages and to remove these stuck readers from the DNA. What happens in healthy cells is that when this reader protein is stuck on the DNA, two proteins, CSB and CSA, are recruited, so they bind to this reader protein that's stuck, and they mark the reader protein with some kind of molecular flag, so to say. And they mark it so that the cell knows, okay, this reader is stuck. I have to do something about it. I have to remove this and repair the damage that has caused this reader to be stuck. So that is what happens. The stuck reader is removed, and then there's room for all kinds of repair processes and proteins to come to this damage and repair the damage. And then ultimately you have the repaired DNA, and then the next reader can come without any problem and transcribe the gene, so make a new messenger of this gene. You can also see this as some kind of, or for example here, on a road where there's traffic, there could be a damage in the road where a car would then be stuck in. First, you have to remove this car from the damaged piece, a part of the road, before you can actually repair the damage in the roads. And only after the damage is repaired, then the next cars, so the next readers could actually go further and drive further on the roads. Well, what now happens in Cockayne syndrome cells? There is, in Cockayne syndrome cells, there's either no CSB or no CSA that's functional. And in either case, that causes that there's no marking of the stuck reader protein anymore. So the reader gets stuck on the damage, but there's no CSB or no CSA, and because of that, there's no marking of this protein anymore. And that way, the reader is not removed from the DNA, and then also the repair proteins cannot access this damage because the reader is really stuck on there, and it really covers the damage too. So there cannot be any repair, and also the reader is stuck on the DNA and cannot be removed. And well, we now more and more think that Cockayne syndrome, the severity of it is not so much caused by the DNA repair, but maybe more by the fact that these readers are stuck on the DNA and not removed. So that is one of the subjects we're really looking into in our research. And indeed, as Joe already explained in the beginning, we're also really looking into what the stress protein HGF3 does in Cockayne syndrome. And I will first explain what HGF3 normally does in a healthy cell. Normally, when you have those readers going over the DNA to make the messengers, this red line coming out, they go over the DNA just normally, and if then the reader encounters such a damage, then it gets stuck, like we saw before. And then CSB and CSA come to the stuck reader, mark the reader so that the reader is removed and then the damage is repaired. But while that is happening, the stress protein ATF3 also comes in on the beginning of the gene, and that inhibits new readers from entering the gene. So new readers are not anymore going over the gene to transcribe the gene. It stops it from going on, because if it doesn't do that, then the readers will be going in and bumping into each other because there's a traffic jam here. So that's what ATF3 does. And then after the damage is repaired, HF3 is broken down again, and then the new readers can go over the gene and transcribe the gene as always. So in this analogy we were looking at before, you could actually see HF3 as these little traffic guys that are stopping the traffic until the damage is repaired. And then after the damage is repaired, they go away so that the traffic can just smoothly go again. Well, this is what it looks like in healthy cells. So after DNA damage, there's a little bit of HF3 that's being made in these cells, and then while the damage is repaired, that HF3 stops new transcriptions from happening. But then after the DNA is repaired, HF3 is broken down again, and there's no HF3 in the cells anymore. What now happens in Cockayne syndrome cells? There also, in the same way, when you damage the DNA, HF3 is produced to stop new readers from going into the genes. But then, of course, in Cockayne syndrome, the repair is never happening. So the damage stays there, and also the readers stay there. And because of that, HF3 is never broken down. So HF3 accumulates in these cells and keep the cell on pause. So they keep stopping new transcription from happening and new readers from entering the genes to make new messengers. And this is a big topic in our research group, to actually find out what the actual contribution is of the fact that the cells are continuously being stopped from transcribing their genes to Cockayne syndrome. Because we actually don't know that, how this contributes to the severity of the Cockayne syndrome, and also how the removal of the stuck reader that's not happening and how much that contributes to the severity of Cockayne syndrome. That's something we don't know for sure. But what we do think or hope is that maybe if we find a drug that removes this ATF3 from the cells again, and then give that way the cell green light again to go transcribe genes again like normal, then that might actually alleviate some of the Cockayne syndrome phenotypes or symptoms. And I will explain a little bit more why we think that might be the case. Because indeed, HGF3 stops new readers from entering the gene, so it stops new transcription from happening. And in this graph, is experiments done by another lab, not our lab, it's publication of another lab, but here they showed that in Cockayne syndrome cells, after you treat them with DNA damage, in cells that have ATF3, which is all Cockayne syndrome cells normally have that, there's very low transcription, and they tested this now for these 10 genes, but there are thousands of genes that are lowered in expression when ATF3 is there. But here for these 10 genes, you see indeed that it's around half of normal transcription is going on in these cells after DNA damage. And if from these same Cockayne syndrome cells, if you then remove ATF3, then this expression is going up again. So all these genes are expressed to a normal level again. And they here removed ATF3 in a genetic way. And they see indeed that the transcription of these genes and thus the production of the proteins afterwards that are encoding in these genes is normal again. And these are not just random genes they chose in this paper. They looked at these genes, and these are all genes that are in some way associated or related to diseases or symptoms that are very similar to symptoms of Cockayne syndrome. So they expect that maybe ATF3 is actually the big cause of Cockayne syndrome symptoms. And that is why we hope that if we find some kind of drug that removes ATF3 from the cells, that might actually be beneficial for Cockayne syndrome. However, I have to stress here that we don't know that for sure. And in this paper, they really think that ATF3 removal helps at least with expressing these genes again. But as I said, we don't know how much of ATF3 or how much of Cockayne syndrome symptoms are actually caused by ATF3 suppression of the cell, and how much is caused by the reader being stuck and not being removed from the DNA. And we don't really know how those two interplay, and maybe there are even other factors involved that we don't know. So we are definitely not sure that this would be the solution, but it is something that we can try, and maybe it would be. So that is why we set out to do this repurposing screen for Cockayne syndrome. And as Joe already very clearly explained, we are using this drug library with around 5,000 drugs that have already been developed for all different kinds of things. And they are all in different stages of clinical development. Some are FDA approved, some are almost FDA approved, and some are a bit earlier in development. But still, the fact that they are already in some stage of development is very beneficial for us, because that way we can skip a lot of steps of drug development. And also for a lot of drugs, the safety profile of giving those drugs to patients is already very clear. And statistically, there is a chance that one of those many drugs could maybe lower the ATF3 levels in Cockayne syndrome cells, and then maybe alleviate some of the Cockayne syndrome symptoms. Well, indeed, Joe approached us for doing the screen, and also she teamed us up with Charles River, a company that's also here in Leiden in the Netherlands, that is really good at performing these kinds of screens. So together we've been trying to develop an assay to perform this screen. Because of course we cannot test 5,000 drugs on patients. That's not really how it works. So that is why we work in our lab with human cells in cell culture lab, that's what we call it. So we have cells, they grow in these kinds of plates or flasks. If you look under the microscope, they are on the bottom of these plates, and if you look under the microscope, you see them like this, and these are human cells that are just growing in this pinkish liquid that's medium with nutrition for the cells. And in the lab, we have all kinds of cell lines, is what we call them. So we have healthy cells, but we also have cells with Cockayne syndrome. And I have to say here that those are not patient cells. So they're really cells that we designed to be Cockayne syndrome cells. So we either removed CSB or CSA from healthy cells, and that way we have Cockayne syndrome cells. And on those cells, we could actually test all those drugs, of course. So for example, if you would put them in such a plate with all these different separate wells and put the cells on the bottom of these wells, we could in each well give one of those 5,000 drugs and see if one of those drugs in the cells cause some lowering of the ATF3 levels. So now we need to find a way to measure these ATF3 levels. And for this, we use green fluorescent protein. That's a protein that's a long time ago been found in jellyfish that are in the deep, deep ocean. And they are green fluorescent. And that is because of a protein they make in their cells that is fluorescent and has this green light. Well, we use that protein and we fuse that to ATF3. And it's for now not really important how exactly we did that. It's a bit too complicated for now, but you just have to understand that instead, in these cells we made, instead of making ATF3 when they need ATF3, they will now make ATF3 fused to this green fluorescent protein. So it's actually ATF3 with a little green light on it. And that's a green signal that we can measure. So now every time a cell makes ATF3, we can measure it via the green light that's attached to it. And if you look at those cells that we made under the microscope, we did this in both healthy cells and in Cockayne syndrome cells. Then you see that if you treat those cells with DNA damage, you get a little bit of ATF3 in those cells and a little bit of green signal here. And that goes away in healthy cells again after the damage is repaired. But then in Cockayne syndrome cells, they also make the same amount of ATF3 after damage, but then damage is never repaired. And that causes a lot of ATF3 to accumulate in the cells. And indeed, you see also a lot of green signal in those cells. So this way we knew our system worked and we could measure the amount of ATF3 that there is in these Cockayne syndrome cells. And now we want to set out and, of course, find maybe a compound that would lower this green signal, so lower the ATF3 levels in these cells again. So we gave those cells to Charles River, and there they have all kinds of machines and robots to really precisely give those drugs in very small amounts in every well on such a place. So what they do there is in such a plate, we put then the cells on the bottom, and if you treat that plate with DNA damage, then every well will become green because all those cells will make ATF3 and will accumulate the ATF3, and then it will become green because that's how we made the cells. And now we of course hope that if in every well we put another drug of that of those 5,000 drugs in the library, that maybe some of those wells won't turn green anymore. Because that could mean that the drug that we administered in that specific well, and we have to look that back then, which drug it was in that specific well, that maybe lowers ATF3 levels in the Cockayne syndrome cells. Well, that's the setup for the screen. And well, we're still in very early stages of the screen, but I do want to show you some early results to give a bit of an indication on what the data coming from this type of screen actually looks like. So indeed, in each well, they put another drug, and now we're looking for those white wells, actually. So the wells that did not turn green. Just to give an indication, they tested a lot of plates to test all those drugs. And then for every well, they measured how green the well was. And they did this in 2 experiments. So I will show you the data in a bit, but now I just covered it with a white plane just to first prepare what it actually will mean, this graph. So the greener a well was in the first experiment, the more to the right the dot will be. And the greener the well was in the second experiment, the higher the dot will be. So every dot in the graph is how green it was in each experiment. And dots that were, or wells that were very green, so had a lot of HF3, will end up here if that was in both experiments. And wells that had very low HF3, so low greenness, and those are the white wells we're looking at, of course, those will end up very low on the left in this graph. And then you also have the possibility that in the first experiment, maybe a well was very green, but then in the second it wasn't, and then it would end up here. And those are, of course, wells we don't really trust because we want to have a nice correlation between the 2 experiments, that 2 times in a row it caused the same effect. So that is why we're really looking alongside this line. And of course, we want to see which compounds now lowered the ATF3 levels. So which are really the white wells in this schematic. And here you see the data that came from the primary screen. And you see a lot of dots here that are in the middle at the 0 point. And that means that in all those wells, there wasn't really a difference in greenness. So they did not become any greener, but they also didn't become less green. There are also some drugs that apparently caused higher ATF3, which could be interesting, but of course for this purpose, we're not really interested in that. What we are interested in are of course the drugs that lowered the ATF3 levels. And those are the yellow drugs in this slide. And here you also see this all these blue dots here, and those were wells that, or cells that were not treated with DNA damage at all. So those cells did not turn green at all. So that way we had a kind of control to see the lower end of how green the cells could become, or how white these wells could become actually. And you see that's actually quite some compounds lowered the ATF3 levels to such an extent that it looked like these cells were never treated with DNA damage in the first place. So indeed, there are quite some compounds that seem to be promising to lower ATF3 levels in Cockayne syndrome cells. But of course, we have to take a lot of steps before this could eventually lead to something really promising. The first step we would have to take is to ask ourselves the question, did the drug really lower ATF3 levels? So did it really do this? Or did it maybe just break the GFP? Did it maybe just break the green light we attached to it? Because that would, of course, also cause a well to be white again. Well, luckily, we have all types of experiments to test really the endogenous ATF3, so without the GFP attached to it in cells. And we could also test this in primary patient cells to see that if giving the drug to those cells would lower the endogenous ATF3 levels. Well, if that looks promising, then we would have to ask ourselves, what is the mechanism behind this? So does the drug really, or how does the drug really lower these ATF3 levels? Does it maybe directly break down the ATF3? So that's just remove the ATF3 itself, or does it maybe work on a higher level, and does it restore the repair in these cells? So does it maybe do something in this process that causes maybe this reader to be removed again, or maybe to help to the repair proteins or something? So that we also have to test. We of course hope that it is on that level and it would really fix the repair mechanism in the cells. And we have also all kinds of assays and experiments to test this. For example, here we can give cells DNA damage on a very small spot here. That's the orange spot. And then we can measure how much repair is happening in that specific spot. And the wider the signal is here, the more repair is happening. And here you see in healthy cells, there's repair happening after you give damage on that specific spot. And then in Cockayne syndrome cells, when you give damage, there's no repair happening at all. And when you measure that, that looks like this. And we can then, of course, see if we treat those cells with the drug, would then maybe the repair be restored again? Well, we have all types of different experiments to test this kind of thing to really figure out what the mechanism would be if there would be really a potential hit here, of course. And if that all looks promising, then we have to see how beneficial it actually is to lower these ATF3 levels when you look at Cockayne syndrome itself, and not only at the model system we now made for ourselves. So we would have to test it in maybe worms or mice models that we have, mouse models that we have that have Cockayne syndrome, and see if indeed this drug would alleviate some of the symptoms that that we see in Cockayne syndrome. So as you hear, there are a lot of hurdles to take still, and we really don't know how beneficial it actually is to lower ATF3 levels. So we really have to see that. But to end on a positive note here, we looked into some other drug repurposing screens that have been done, and we asked ourselves, is this approach, has this worked before? And I found a study from a lab in the USA, and they did a similar approach to try and find a drug that maybe would help in the MUC1 kidney disease. And how I understand it, in this disease, there is this misfolded protein. And normally misfolded proteins would be cleared out by the cell, would be thrown away and destroyed by the lysosome. That's some sort of garbage disposal of the cell. And misfolded proteins should be brought to that so that it's destroyed in the cell. And in this disease, apparently that does not happen to this specific misfolded protein. So that misfolded protein accumulates in the cells, and then it causes cell death and eventually kidney failure. And they did a similar approach for this disease to find, with drug repurposing, find a drug that might help in this disease. So what they did, they screened all these compounds in another assay that they developed specifically for this disease. And they tried it on different doses, and like us, we now tried this primary screen on 2 different doses. Then they got some hits, like we do now, or yellow dots in the graph. And then they tried that on all kinds of different doses, and they checked the activity and the specificity, the viability, the toxicity, and all kinds of stuff. And in the end, they ended up with one compound here, this compound, which was initially developed as an anti-hypertensive, so to lower blood pressure, I understand. But they found that in their disease, they tested it first on cells with the disease, like we're doing now. But in the end, they tested it on mice that have the disease. And without treatment, these mice have this misfolded protein. They stained that in green here in the kidney. But if you treat the mice with this compound, then the misfolded protein is removed from the cells, and they only have the correctly folded protein in there still. And they also found out a mechanism on how this works. Apparently, this compound binds the misfolded protein and guides it back to this garbage disposal of the cell. And this accumulating toxic compound is then removed from the cell, and clearing out the kidneys. And this is quite a recent study, so I couldn't really find how far the clinical developments of this are, but I thought this really looked promising though. But then I also have to note that in our screen, we are still at this phase, so we have a lot of steps to go, and we really hope that something here like this compound would come out, like it did for this screen they did, but we are still here. So there's a lot of steps to take still. But yeah, for that, with that, I would like to end here my presentation. I want to thank, of course, everyone in the lab and Martijn for all his guidance in this project. And of course, everyone at Charles River for the nice collaboration and all the great transparent communications that's going on. And of course, Joe, for bringing us together and for organizing this whole project and organizing this webinar as well, and all the great input you gave until now. And thank you, and I would be happy to take any questions together with Martijn and Joe, of course.
Yes, thank you so much, Paula, for that very comprehensive presentation. So essentially, we're trying to figure out how to kick those traffic guys to the curb.
Yeah.
So that's really sort of the meat and potatoes of it. I also just want to, before we go into questions, just to give a specific thank you to our donors. Riaan Research Initiative is able to fund this work because of our donors. And while the costs aren't as astronomical as developing a drug from scratch, which can cost $1 billion, it's still the cost of buying a house. So, these are very expensive programs. So thank you so much to our donors who support this work and are able to make hope possible for the Cockayne syndrome community. I'd like to open it up now. If anybody wants to ask questions, just to raise your hand and speak, please do so, or you can type it into the chat box. I don't think we have anything open right now. Oh, we just got one. Thank you for such a nice presentation. Considering its connection to DNA repair, do CS patients also develop cancer? Martijn or Paula?
Yeah, that's a very valid question. I think they don't, but I think Martijn can give a little bit more of an elaborate answer on that.
Yeah, no, definitely not. No. So I think the one way of looking at this is that DNA damage can have two different, pretty opposing outcomes. So on the one hand, it can be very toxic to cells. And if that's the case, then that can lead to neurodegeneration. That's clearly the case in Cockayne syndrome. And the other outcome is that it can cause mutations. So there are, let's say, mistakes made during the repair process or in other processes that try to bypass the DNA damage. And this then can lead to cancer. So, and that's, for instance, seen in xeroderma pigmentosa. So that's another syndrome where DNA damage is affected that clearly leads to skin cancer induced by sunlight. But that's not at all what we see in Cockayne syndrome. So these patients do not get cancer. No.
Okay, we got another one. Is the aim of the medication to reduce the ATF3 rather than to remove it fully? Good question.
Yeah, that's indeed a very good question too. I think we don't know for sure what the effect would be, and if we would have to remove the ATF3 altogether or whether lower it would already be beneficial. And those are all things we have to test when we find or if we find compounds that would actually lower it, and to what extent is it beneficial, and how much removal of ATF3 is needed. We don't know it yet, I think.
As we wait for any more questions, one just popped up. Two just popped up. Really exciting work, Paula and co. Does ATF3 upregulation CS cells vary between different cell tissue types? That's from Chris Carney. Thank you, Chris.
Yeah, that's also a very valid question. I'm actually not sure. I don't know if Martijn knows this.
No, I think that's a great question. And I think to address that, we would have to make an ATF3 GFP knock-in mouse and really expose it to different types of damage, and then, looks tissue specifically whether there's an effect or not. So that's something that's not known at the moment.
I do want to, we had some conversation in the beginning of the project about whether ATF3 is expressed in the brain, and there is some evidence that there is. Would you be able to speak to that a little bit more, and then I'll jump into the rest of the questions?
Yeah, so it's definitely expressed. The interesting thing also is that if you look at the genes that basically are downregulated also in the cells that we've made, but also in other cell models, if you compare that to the analysis that have been done on brain tissue from Cockayne syndrome patients, they match pretty well. And a lot of these genes, there's around 4,000 of them. They have a binding site, so let's say a region that's recognized by ATF3 very early on in those genes. So I think all the evidence is there that ATF3 is one of the key players that is downregulating these genes also in the neurons of CS patients.
This is the next question that's relevant to this. So I'm going to skip to that. Is if you remove it entirely, I'm assuming ATF3, will it then cause the traffic to cause a pileup as the DNA has not been repaired?
That's also a very accurate question indeed. It might, yes, but we think that the cell being constantly, or the transcription being constantly passed by ATF3 is more harmful for Cockayne syndrome than the piling up of the readers on the DNA would be. So that is why we think this still could be a beneficial way, but we also have to check that whether if we find a compound, whether that actually happens, or maybe something in a higher level prevents that even from happening.
And to add to that, so in these experiments where ATF3 has been removed, so in that case, that's genetically by just taking out the gene, you see that a lot of these genes that are typically expressed at lower levels in Cockayne syndrome cells now are upregulated again, so they're expressed at higher levels again. And I think that's a clear indication that it's not the DNA damage itself that's causing this reduction and this, the blocking, but it's actually also in genes that have no DNA damage at all. ATF3 will shut them down. But so we don't know really. On the one hand, you could indeed argue that because there will be DNA damage in those cells, that if you don't have ATF3, there will be more of these readers that will pile up and accumulate there, and that's certainly possible. But on the other hand, there are now many genes that are not damaged at all, and they're kind of shut down as a precaution. And I think we want to prevent that from happening.
Next question is from Janie Hughes. Janie runs Amy and Friends in the UK. Hi, Janie. Jane, are the drugs used approved by EU as well as FDA? A very good presentation. Thank you.
I'm not sure, actually, Martijn.
No, me neither. So they are for sure FDA approved, but we would have to really check. But I think that we can do further down the line, because we started with 6,000 compounds. Maybe there's someone here from Charles River that knows much better than we do what the answer to that question would be. So I think now we are close to 120 lead compounds that are worth following up, and then at Charles River they're doing further potency testing, and we're reducing that number. And I think when we're at that stage, we want to know exactly what the compounds are, what's their mode of action, what pathway they work in, and then also are they also approved by EMA and by other European organizations as well. So that's definitely worth exploring that.
It's my understanding that, and if anyone wants to pipe in, they can, but it's a combination of FDA approved drugs, but also drugs that are in clinic that have been found to be safe, but are not necessarily considered effective for any other diseases at this point. So likely there's going to be an overlap. But whether the exact hit list is going to contain drugs that are EU approved and FDA approved, those are some outstanding questions. I mean, there's other things that we have to think about is whether the drugs will be, have they been tested in children? What is the route of administration? Are they pills? Are they liquids? A lot of our children are in feeding tubes or can only take liquids, intake liquids by mouth. So those are some of the things that we have to think about as well as part of this. The next question is from Sophie. Sophie says, is there a special diet that could help to reduce the radicals intake for our children?
Yeah, so unfortunately, no. I mean, that would be, of course, great. The thing is that I think, food is already quite a big issue in these children. I mean, because they are small and feeding can be an issue. I mean, I think they kind of restrict themselves in a way, right? So I think it's very important to discuss this with a dietitian. And I mean, that's not our area of expertise at all. But in this case, whatever food you basically consume, this will lead to oxidative damage and damage just by metabolizing the food that will cause DNA damage. And I think that's inevitable. So of course it's important to not overfeed, but I think this should always be discussed with a dietitian. And I mean, also, at least I know from Amy and Friends in the UK that they collaborate with a lot of dietitians that know a lot about this topic. So then I would definitely get them involved before making any changes to the diet. But I would never do this, on your own based on some of this work, for example.
Sophie, we can refer you to Amy and Friends if you're not yet connected, and they can connect you to, there's a CS clinic that they work with, they collaborate with in London to help children with this disease. And they have definitely experts and dietitians on staff. They can, and also other parents, if you're not already a part of the support groups, that can answer some of these questions. All right, anybody else? I'm going to ask, I think this has been sort of already addressed, but do we think that this will help patients both with CSA and CSB mutations? We know the majority of CS patients have a mutation in CSB/ERCC6.
Yes, we would expect so, because CSA and CSB really work as this pair to recognize the stalled reader and then also to mark the stalled reader to be removed. So if either one of them is not there, this process doesn't work anymore. And it also, of course, depends on which level the ATF3 removal would be caused. So we do need to look into that. We do expect that anything we would find would work in both cases.
Any other questions? Again, if you don't want to type, just raise your hand and I'll unmute you if you want to ask. We have about 6 minutes left, so we're good on time. Okay. I think that's everything. Thank you so much, Paula and Martijn, and everyone who has been able to join us today. We did record this presentation, and I know it was very dense and there was a lot of heavy material. So if anybody wants a copy of it, we'll send it out to the participants who've registered, but we'll also, you can reach out to us at info@riaanresearch.org, RIAAN research.org, or reach out to us on social media. And we're on Twitter, Facebook, Instagram, and we will make it available to you. There's one more question. This is from Gianna Dyer. How long do you project all of this to take? Sort of the $1 million question, right?
Yeah.
Yeah, that's a very valid question. Question too. Yeah, it's definitely gonna take some time. And actually, I'm not really sure how long we're talking about here. I think we've now been working on this for almost a year, I think, to develop the essay and then get into this first stage of screening. And there's a lot of stages of screening we still have to go through, I think. And then after that, of course, finding out what is actually happening with these compounds that might be potential hits here. That's going to take quite some time.
Yeah, so maybe to chime in here, I think you can actually think about it in different ways. So let's say the fastest way would be now if we find something that is already FDA approved in principle, it could be subscribed off label, so that could go very fast. That's also why we, decided to go for this, FDA approved list of compounds. But of course, if we find something that might be promising, but it needs to be adapted, then it's a different story. Then maybe we have to go through approval again. And if we also want to test it in worms or in mice or in these type of organisms, that will of course take time. But I think it doesn't have to go in that specific sequence. I think in principle there would be other options to already test it, maybe in a limited number of patients, but that we then really have to discuss with clinicians and doctors involved. And of course, Joe has a lot of connections in that area, so I think that then can be discussed what the possibilities are. Because that's the beauty of now starting with these compounds. If we would have started from, let's say, a lot of different chemical structures that we don't know what they're doing, then if you find something, you would have to go through all the steps, through the worms and through the mice and everything. But I think we can kind of skip some of that because we already know that these drugs are safe.
Yeah, and to give you an example, there was another rare disease drug repurposing program where they discovered that an asthma medication, something as simple as an asthma medication, was able to help alleviate symptoms for their disease. And that's easy. It's already approved. It's known to be safe. So those are easy things for a doctor to potentially prescribe off-label. So not for its original intended use, but for a different use. But then there's other factors we have to consider, like insurance costs. Will insurance cover it? How much will it cost to get access to the drugs? So, there's best case scenario, and our goal is, as an organization, which is why we were founded, is to move with as much urgency as possible, as much expediency as possible. And that's what we're trying to do. But we also have to keep in mind that we want something that's safe, and in addition to being effective. So there's a lot of variables at play, but we're trying to move as quickly as we can. And with your support, we're able to do that. So any other questions? All right, we got a thank you from Janie again. Thank you so much. And also thank you to everyone else. I think that's it. If you have any other questions and you want to ask them privately and you want to connect with Martijn or Paula, please reach out again. That's info@riaanresearch.org, or connect with us on our social media. Thank you again and have a wonderful day. Take care. Give a big hug to all of your kids for us. Bye.
Bye.
You.